Hello everyone. In this tutorial, we will talk about how to download gene expression or copy number variation or genomic sequencing data using a very friendly, user-friendly platform that is ArrayExpress. This ArrayExpress platform is a package of EBI Amble Institute, and a lot of things can be done using this platform, right? So let's go and see how it works. First of all, you can just search ArrayExpress and then you can find the link of EBI website, and here you can see this is the main GUI of ArrayExpress, and that contains all the information related to functional genomics data, right? There are 73,751 experiments, more than 200,000 assays, and then almost 60 terabyte data. Let's say here I'm searching breast cancer and I want to look for the information related to breast cancer available on EBI, ArrayExpress, right? So this is the excision number, this is the title, and the title of the study, which was uploaded, the dataset was uploaded here. Then other titles like microRNA regulate expression and mRNA sequencing of HC19 and so on. Then there are types like DNA sequencing data, chip sequencing data, RNA sequencing data, RNA sequencing of total RNA, microRNA profiling, high throughput sequencing data, transcription profiling data. So there are so many types of data sets available here. Then organism type is also available like human, mouse, Drosophila, and so on, right? And next, the number of assays performed is also available. Like one study has 86, one has 15, one has 88, 16, and so on. So these are the number of assays performed for each study, and the release date is also available. And then there are two types of datasets. One is the processed data and one is the raw data. So some studies have both the processed data and some studies have only the raw data and so on. Here you can see that there are three types of links. One, you can link from that chain tells you that you can go on the website from where you can download, and this arrow sign shows that you can directly download from here, right? So if I click any one expression number here, you can see that it will take us to the detail where you can see that this is the title, then status, organisms, total number of samples, protocol used, and then at the end of that you can find two main things here. This is a bit shorter data set. Here you can see only investigation description and the sample data relationship are available. But if I look for some comprehensive data or large data set, you can let's see, I've just sorted from largest to smallest and looking for any data set. Here you can see this one, transcription profiling of array of breast cancer samples to define breast cancer subsets. I just clicked it. Here you can see that this is the title and the description, and then the study authors. Now this is investigation description. You can just click save it. I'll show you what does that mean, that investigation description file. Then the sample and data relationship file. These are the two files which will tell you the details about this experiment, right? So if I go and open these files and show you the details, okay, so later, and then raw data and array design data, I'll tell you. The raw data actually is the data of raw data set which you have to process all, and the array design is the description of that file. So here if I click the investigation description file, so you can see that all the description of the investigation description, like the authors, time of the study, title of the study, other protocols, and the institute which submitted this. So the basic details of the investigation of the study are all available here. And the next thing is the sample data relationship file. This is important because when you are analyzing the data, you want to see the description. For example, here this is the sample number first, and then the whole organism, then this is the data set of breast carcinoma and other details as well. For example, the patient is female, and so on. So depending on the type of depending on the experiment, there are some more details compared to the every study has different set of details. And again, then array design is actually the design of this array which is used. So you can click here, you can click the raw data and you can download all the files individually as well and as collectively, and so on. So this is the design actually is of the FE stands for FE metrics. So this is FE metrics platform, right? So these are all the details. So you can download the raw data and also the processed data as well. So now if I want to filter the results, for example, I want to look for any specific type, for example, Homo sapiens, I only want to see the data of type and RNA assay, and then only the array assay, right? And I also try to select only one array type. For instance, here, let's see, I click this 133 plus array, right? Now, okay, I just click the filter. One more thing, that if you want to see for only ArrayExpress data or the other data sets like from Geo or NCBI, I just clicked it, and we cannot find any match for the experiment. So it means that the descriptive filter we set were no dataset was available against that. So now I am just looking for if I should whether I should select any data or I just keep it that way. Let's see. Okay, let's see if we just keep it that way and I just go for the RNA assays. So now you can see that all the technology datasets are available here. So you can filter by experiments as well. Right? This is highlighting the main things we want to see. So let's see if we want to search specific search in the search box. For example, we want to look for breast cancer data with sample count or assay count of 1 to 200. So I don't want any dataset which is greater than 200, right? So so this will give us a specified set of datasets, right? Let's wait. I think the internet got a bit slower here. I just entered it and let's wait for a few seconds. Now, yeah, now it's ready. So here you can see that now all the datasets are, now this is 15, 88 and other so on, and if I just sort it by largest to smallest, so here you can see that when I just click the largest to smallest, this will show the maximum number of assays will be 200 because my filter is between 1 to 200. Yeah, now here you can see that this 200 actually means that the maximum number of assays because I selected 200 here, right? I can change that with the other sample count as well. Sample count actually means that sample count is different from assay. Assay is actually an experiment or you're performing, and that on the number of samples we want to perform means different. That total number of samples will be different from the total number of assays, right? So here I've just searched sample count if you're interested in only samples. So here you can see that now the sample count is 177, 95. So here you can see that 95 is actually the assays, but the number of samples may be more compared to the assays. So just that is here I just concluded. So it means that now you know that how to look around for the data on ArrayExpress and how you can download the relevant information and how you can process that information is also available on my YouTube channel. So I think that's enough for today. Thank you. Allah Hafiz.